There are various plant diseases caused by fungal infection, such as powdery mildew by Ursidaceae family, Dodge disease by Ceratocystis olivini, corn smut by Ustilagomegalis, and blood knot by Pleuroitia morbosum. Moving on to the literature review, currently there are 300,000 species of microalgae. However, only 10% of it have been documented. Microalgae thrive in diverse natural habitats and can rapidly adapt to varied extreme conditions, resulting in the potential in producing metabolites with unique structure which may not be found in other organisms. From Chlorella vulgaris, Beapi et al. 2018 succeed to extract 6 bioactive compounds that able to fight against 6 phytopathogenic fungi. From here, diethyl ether was most efficient against the fungal, followed by methanol and acetone. Based on Schmidt et al. 2022, amphinidols extracted from Amphididium cattarae had shown lower environmental footprint, reducing toxicity compared to synthetic fungicide. In the same study, it was revealed that S. obliquus exhibit the highest inhibition against target fungus, up to 32.01 plus minus 4.82%, and highest biological activity against bacteria, fungi, and cancer lines is possessed by the diethyl ether extracted from these microalgae as stated by Mares et al. 2019.
In the first part, it includes sample collection, purifications, and identifications of isolates. Firstly, the sample is collected in container wrapped with aluminum foil. In the lab, the sample is filtered by using glass filter. Then, the serodilutions of filtrate is performed to isolate microalgae in blue-green BG11 media via spread plate and incubate for 2 weeks. Then, the microalgae strain is further isolated by straight plate in petri dish containing BG11 media. Then, the colony formation is confirmed and the colony is removed by using wire loop and placed on a slide. Lastly, the morphology of microalgae is observed under microscope, then match identification code with algae compendium present in freshwater algae in British ISIL to compare the morphological characteristics of isolates. The wastewater is selected in this study to obtain microalgae because microalgae grow rapidly in wastewater environment as it can thrive in harsh conditions. This is due to the abundance of carbon, nitrogen, and phosphorus sources in wastewater which serves as nutrient for microalgae. Also, the production of wastewater is predicted to increase globally by 24% to 51% by 2030 and 2050 respectively. So, the use of wastewater to isolate microbial declines in terms of cost in developing biofungicide. It is important to provide microalgae with culture conditions that mimics its natural habitat. BG11 is usually used to isolate and cultivate the majority of microalgae as it provides all essential nutrients. Additionally, it has been widely utilized to identify across diverse species. From the identifications of microalgae, we are expecting the isolation of Chlorella species and Corysum species. As reported by Abergi, both of these species were identified from the industrial wastewater with similar media, whereby Chlorella species appear as spherical green algae and lack of flagella, meanwhile Corysum species appears as dark green colony and oval spindle in shape. Another finding also reported both of these species can be identified from industrial wastewater with different media. Additionally, it was found that Chlorella sorokiniana and Chlorella vulgaris can be isolated from wastewater.
To assess antifungal activity, we extract bioactive compounds from Chlorella species and Corysum species using solvents selected based on polarity. Diethyl ether, a nonpolar solvent, extracts lipophilic compounds like lipids and nonpolar terpenoids. Methanol, a polar solvent, extracts phenolics, flavonoids, and polysaccharides known for antimicrobial properties. Acetone is included to cover a broader polarity range. Maceration is used for extractions due to its simplicity and efficiency. Biomass is soaked in each solvent for 3 days and the extracts are filtered with Whatman No. 1 filter paper and evaporated to obtain crude extracts. These residues are dissolved in 10% DMSO to create stock solution for antifungal assays. Antifungal activity is assessed via the ARCA well diffusion method, chosen for its simplicity and clear inhibition zone visualization. Fungal pathogens are cultured on PDA and spores are inoculated using a sterile cotton swab. Wells are created and loaded with 100 microliter of extract at different concentrations, alongside positive and negative controls. Plates are incubated and inhibition zones are measured to determine antifungal effectiveness. Based on previous studies, Chlorella species was selected for characterization due to higher antifungal potential, while Carasium species lack sufficient research. For compound identification, HPLC and GCMS are used. HPLC analyzes non-volatile phenolics and flavonoids from metallic extracts. Samples are separated and the compounds are identified based on retention times compared to standards. HPLC is chosen for its high resolution, sensitivity, and ability to quantify key antifungal compounds. GCMS analyzes volatile, lipophilic bioactive compounds from diethyl ether extracts. The sample is vaporized, separated by a capillary column, and identified via mass spectrometry against reference libraries. GCMS is preferred for its high sensitivity and accuracy in detecting low molecular weight compounds.
Secondary screening of another compound using MIC assay can be done to evaluate antifungal potential. We pinpoint 3 key compounds, which are caffeic acid, carbapenem, and denise, based on their reported antifungal activity, with carbapenem showing the lowest MIC value at approximately 80 microgram per ml against Candida albicans. Amphotericin B will be used as positive control in this experiment, while solvent without any compound of interest added will serve as negative controls. The procedure will involve preparing stock solution, performing serial dilution, inoculating fungal strain of C. vulgaris, and incubating under optimal condition to determine the lowest concentration that inhibits visible fungal growth. For the biofungicide formulation, we can mix the active compound carbapenem with suitable carrier. It can be water or oil-based solution, and we can incorporate stabilizers or surfactant, which helps in enhancing application efficiency. And in order to test the effectiveness, the formulated biofungicide will be applied to fungal infected plant models to assess its antifungal activity. And in order to do so, we have to validate it by comparing it to controls, which the positive control we will use commercial biofungicide such as amphotericin D, which to compare efficacy, and negative control using the plain solvent without any carbapenem added to ensure that any observed effects are due to the active compound.
For validation, the biofungicide will be tested under real agricultural conditions through small plot field trial conducted outside the laboratory to assess the effectiveness and efficacy of the biofungicide. Randomized complete block design RCBD is suitable for field trial with 3 replicates per treatment. In this trial, selected plants with fungal infection disease such as powdery mildew infection will be sprayed with the formulated biofungicide 3 times at 7 days interval as applied in the study by Shin et al. 2017 and Bikash et al. 2024. A control group received no treatment while another group is treated with biofungicide. Some studies also compare the biofungicide with the existed biofungicide in the market to make sure it meets industrial standard. Next, disease incidence DI will be assessed 7 days after the final application. The formula is control efficacy equal to 100 minus 100 multiplied with disease incidence of treatment divided by disease incidence total. Lastly, Tukey's HSD test was applied at P equal to 0.05 to compare the treatment across block and the significant difference between the treatment and the control.
For justification, we choose small plot trials as they allow precise disease monitoring and biofungicide efficacy while mimicking real agricultural conditions. RBCD was chosen for this field trial because it is expected to minimize environmental variability to ensure that the differences in DI are due to the biofungicide treatment rather than external factor. And for expected outcome, we expect that there will be a significant decrease in disease incidence DI in treated plant with biofungicide than the control. The biofungicide should also show a comparable or superior control efficacy to the existing commercial biofungicide.
In conclusion, the development of a biofungicide begins with wastewater sample collection, filtration, and serial dilution before isolating macroalgae. The isolates are then screened for antifungal properties using well diffusion assay. GCMS is used to characterize bioactive compounds, followed by MIC determination and formulation. Lastly, validation is conducted through small plot field trial with RBCD. The limitation includes the difficulty in identifying and characterizing bioactive compounds due to the complex nature of macroalgae extract. Additionally, optimizing the biofungicide formulation is challenging as the concentration of bioactive compounds must be carefully balanced to prevent harming the plant or crops. These are the references we use, and that concludes our presentation. Thank you.